Red blood cells are mainly, their main cytokine is called erythropoietin. I'll show you a bit about how that's controlled in a second. And platelets are a thing called thrombopoietin. And just as an example, it's amazing how this process works. And the two examples I want to show you is erythropoietin. If I lose some blood, let's say I donate a unit of blood or cut myself and lose some blood. What happens is that there are less red blood cells in my body, in my blood. So there's less ability to carry oxygen around the bloodstream. And there are special cells in the kidney, and the kidney gets a lot of blood flow. About a fifth of the blood flow goes through the kidney, so it's a good place to have something to sense how well the blood's carrying oxygen. There are sensors that can sense this. They sense there's not as much oxygen coming through, and in response to that, they secrete this hormone called erythropoietin, and that travels in the bloodstream to the bone marrow, where the bone marrow producing cells are activated. So it binds to receptors on those bone marrow on those blood producing cells, which switches on certain genes and they start dividing more rapidly. Okay, so that's how, and you know, the cyclists all cheat by sort of abusing this, by injecting erythropoietin to get their blood count a bit higher because they think it gives them an advantage. It probably does, but it's not good for them. Okay, so this is how red blood cells are controlled largely. Platelets is a similar thing, but it's quite different. So instead of switching on and off, the liver just produces this thrombopoietin, this hormone, at a constant rate. It doesn't change. It doesn't matter what you do, that just gets produced. And that thrombopoietin gets into the bloodstream, and the platelet cells, which are the clotting cells, bind that. And it doesn't do anything to the platelets, it just sucks that hormone up. But a little bit gets through to the bone marrow, to these megakaryocytes, the cells that produce platelets, and stimulates them to produce more platelets. And it's very elegant because if you produce more platelets, there's more platelets here that suck this up, so less gets to the bone marrow, so it sort of switches it off. If you've got less platelets, more gets through to the bone marrow, which stimulates these cells to produce more platelets. So it's a nice sort of feedback loop. And it's quite different from erythropoietin, where the level of the hormone goes up and down. Whereas here, the hormone's the same, it just depends whether it's sucked up by the platelets in the blood or not. So that's the sort of background. Now, I guess the question is, what could possibly go wrong? And this is actually, we had a patient on our ward who had a calendar with these cartoons, and there's 12 of them. I've used 5 of them to talk. My aim is to use all of them. They've been laminated, put on the wall. And here we have the TV presenter with the informant for the organised crime syndicate who's going to talk to him in the dark. And of course, what goes wrong is someone turns on the lights, so his face is. So, you know, plenty can go wrong, even when you least expect it. And the key thing is that you can get mutations in key genes in these cells in the bone marrow, which makes cells behave badly, behave inappropriately. It makes them divide out of control sometimes. It stops them from growing up sometimes. If you switch off the wrong genes, or you damage or change the genes in your cell, you can have cells that don't grow up properly. You can have them dying before they should, or you can have them not dying when they should. And all these things lead to problems. I'm going to show you examples of that. So I want to briefly talk about mutations, because there's a couple of them. That we can have, you know, I mentioned the DNA is in this series of chemicals, bases, and they have this ACG T pattern, and you can have a change in the base which alters that sequence, and that's what we call a point mutation. So a single base, and what that sequence does is it tells the machinery of the cell to make a protein with these what we call amino acids. There's 20 of them, and this code tells which order the amino acids are put together. And if you change this code, as has happened here, so instead of a UGC, which codes for this yellow-orange triangle to be incorporated into the protein, that actually tells it to stop making the protein. So you've got an abnormal protein. So point mutations change the code and change, either stop the protein being produced or change the nature of the protein being produced. Okay? And the other group is called translocations. You know, I mentioned how the DNA, when cells divide, is organised into these chromosomes. Occasionally, a bit of one chromosome lines up with a bit of another chromosome, and a big chunk of DNA swaps. And you can see this here is this is the normal chromosome 22. You end up with this little short one. That's called the Philadelphia chromosome. And what happens when this occurs, you get bits of DNA, like this bit called BCR on chromosome 22, and this bit called Abel on chromosome 9, who are they're nowhere near each other normally. They may as well be on different planets. They get stuck close together, and you get this abnormal gene, which doesn't exist in normal cells, and that abnormal gene can drive proliferation. This is the basis for chronic myeloid leukemia. You get this abnormal BCR-Abel gene caused by these big chunks of DNA swapping on these chromosomes, which then drives the cells to proliferate out of control. I'll show you some more about that in a minute. So I want to develop, we're getting towards the end, a model for the bone marrow, because I've talked a lot. I guess hopefully this will reiterate what I've talked about, but in a way that's perhaps a bit easier to understand. And I guess the bone marrow starts with a stem cell. and undifferentiated. This stem cell can become anything, okay? And well, I'm not going to tell you how it's delivered, but you know, let's just say the stem cell's there. So here's our stem cell. That stem cell is not much use. It can't function to do anything, but everything that's useful starts off as one of these. Everyone in this room starts off, okay? So stem cell's like a newborn baby, okay? So it's undifferentiated. And in the bone marrow, we have these stem cells growing up into these immature bone marrow cells, starting to work out what they're going to do with their lives, but still not much use, okay? And out in the community, we have the mature blood cells. And you see I'm politically correct. We've got a female doctor and a male nurse. We've got a soldier. You know, we've got the miner. So we've got the lawyer. I'm not quite sure what he does, but we need him anyway. And, you know, the food producers, etc. And this is the mature blood cells. With all have their roles, okay? And, you know, without these don't exist. Without these don't exist. But these actually are no use, and neither are these, okay? Just like human beings. And this is what does this look familiar? We've got the stem cell and these sort of various immature cells that are in the bone marrow growing up, differentiating into these more mature cells. And remember I mentioned this sort of lymphoid cells down the side here. I haven't got them in the model, but they're actually cells. Some of these immature cells travel quite early in their life out to the lymph nodes and the thymus, which is a gland up in the throat and the neck here, and the spleen. And it's like a military training academy, because the lymphocytes are part of the defence system. So you have these sort of baby soldiers, it's like boarding school, and they grow up into mature soldiers, which then can go back into the bone marrow and function. So that's another part of our defence. So these are the sort of foot soldiers. This is like the officer training school, I suppose. And so I'm now going to start with some diseases. What happens if your stem cells fail? Okay, so if you lose your stem cells, you've actually lost everything. Although stem cells are no use out in the blood, if your stem cells come under attack, you've lost everything. And aplastic anemia is the disease where you lose your stem cells, okay? And it occurs usually because our immune system, for some reason, attacks our own stem cells. So if you think of our stem cell delivery mechanism, and our own immune system gets it wrong, okay? And so here's our picture. This is normal, okay? You take out those stem cells, everything disappears, okay? And that's a bone marrow from aplastic anemia. Nothing there, okay? Because everything's gone. Now, that sounds really serious, but we can treat this. We can treat it by suppressing the immune system, making it leave the stem cells alone so they can grow up and this all reappears, okay? But that just shows you how crucial stem cells are. But a stem cell cannot carry oxygen around the body. It can't fight infection. It can't stop you from bleeding to death. So what happens if you have uncontrolled proliferation? So let's say that instead of this all being controlled, this sort of expansion here, so we get lots of these people out of that stem cell, let's say we lose control of that and we just get uncontrolled proliferation. In particular, if one group, like the army, takes over, okay, we have sort of completely unbalanced, we get excessive population, an unbalanced workforce and instability. And this example is chronic myeloid leukemia. Okay, so we have an early cell which, instead of dividing when it should do, stopping when it shouldn't do, just loses control and just divides. You still get lots of mature cells, but it's completely unbalanced and you get into trouble reasonably quickly when that happens. And this is the normal bone marrow again, and this is a chronic myeloid leukemia bone marrow, where it's absolutely packed with cells. Okay, still producing mature cells, but you get into trouble over it without treatment within a few months if this occurs. Now, we talked a lot about these cells growing up, you know, so what happens if that fails, if differentiation fails? You get this, okay? You've got your stem cells there, but you end up with this. You end up with a whole load of 3-year-olds who are useless, but what's worse is they can reproduce. So I always talk to people about the household full of 3-year-olds who are actually having more 3-year-olds. I mean, that is chaos fast, okay? So a failure of differentiation causes trouble quickly, and that is acute leukemia, okay? Acute leukemia, we know, develops quickly and causes trouble quickly. And it's mainly, I mean, there is excessive cell division, but the main problem is the cells won't grow up. So you rapidly lose mature cells, and your bone marrow fills up with these reproducing 3-year-olds, okay? And that's an example. So you've got this normal blood with the nice mature red blood cells and nice mature neutrophils. These are acute leukemia cells. They're immature. That nucleus is very big and open. These cells are dividing, but they're useless. Okay? Although they are destined, or should be destined, to become nice mature cells like this, they can't grow up and they're useless. And they fill up the bone marrow and stop the bone marrow producing useful cells. So that's acute leukemia.